and then more recently moving to NIAB, where he's head of data sciences. He also holds a part-time professorship at the Nanjing Agricultural University in China, and is an honorary professor at UEA. Without any further ado, Xi, I'll hand over to you.
Thank you very much. Can you hear me well on the back? Can people online can hear me? Fantastic. Okay. So thanks for Rob's invitation. It's a great honor to come here and learn from the research conducted at James Hutton Institute. My talk has been divided in two parts. One part is about demonstrate the capability from our side, what we can do at multi-scale plant phenotyping and crop phenotyping, not only in the greenhouse but also in the field. And the second part is about trait analysis. How can we get traits from large-scale imaging, and how these imaging-based data can be feeding to trait analysis and eventually getting to the genetic mapping and also gene discovery. Right, okay. So first thing first, this is the Cambridge crop research. I think we're using a drone to mapping the 3D map it. And you can see right here that the entrance of this. Can I have a mouse? I can't have a mouse. Isn't there a way that I can show?
Yeah.
Like this?
Yeah.
Like a pen, okay.
Yeah. You should be able to get that.
Like this one.
There you go, yeah.
So basically, okay, marvelous. Thank you very much. So this is the entrance. I hope in the near future you guys can come over to Cambridge Crop Research and then NIAB to visit in my lab and visit other researchers, leading researchers at NIAB. And this is the team, and obviously the team has been changed. Jeremy has gone to industry, competing with these bloody Cambridge companies, but coming back, we have been getting a lot of fresh blood from the universities as well. So we start to work a lot on trait analysis in field and in greenhouse. What do I mean multi-scale plant phenotyping? As you can see from this slide, what we mean when we're talking about multi-scale plant phenotyping, I mean from different levels, from cellular level to the tissue level to organ level, gradually going up to the macro, like plot level, and the population, field population level. And we have been generating a range of methods to capture data from these multi-scale levels. And then computer vision and AI have been one of the major breakthrough that my lab and department have been investing on. So you can see from here, at the cell level, if you want to look into stomata closure, if you want to look into disease infections, try to rank it at different classifications for severity level, or flowering, you want to understand time series-based flowering analysis on wheat, rice, cereal crops. Even you're using a smartphone in the field and taking an image and then tell you how many spikes or how many panicles within the unit area. And if we're going up beyond the plot level, if we want to study the breeding plots to generating large-scale trait analysis directly from a drone flying, this can be all analyzed. Not only, I'm using AI as one catchy word, but there's a range of methods we can develop and extract the meaningful information from the large data we're facing from our daily research. This is the contribution from my Chinese and UK lab, mainly from the UK lab, and at different levels. So you can see previously when I was at TSL, we've been publishing a lot on the cellular phenotyping, high-throughput, high-content screening system to study plant pathogen interactions. And when I moved to join the center and the TGAC, we've been working a lot on the field-based analysis to understand how we quantify disease, how we quantify seed germination, how we look into organs that are particularly interested by scientists at the Norwich Research Park. And more recently, when I started my professorship at Nanjing Agricultural University and also NIAB, field-based large-scale have become much more important. Through this particular approach, we've been looking to traits. So what kind of traits are can be utilized to assessing the nitrogen use efficiency, what kind of traits can be utilized to quantify the stress level, as well as how the key yield components or yield or genetic gains can be measured year by year, are the things that we've been paying a lot of attention. Right, so as you can see, as I said, the first part of my talk is more about what kind of work, what kind of capabilities that my department can provide and can be collaborating with a broad community in plant research. So the first part is cellular phenotyping. This one has been done previously on using the Opera system. Opera HCS system basically provide 96 wells when you mount your leaf tissues, mainly Arabidopsis and Nicotiana pensemiana, onto the 96 wells. You can image them using random region of interest, and eventually what you're getting is a huge amount of data for the cell, at the cellular level. For example, these are epidermal cells, and then you can study things like plasmodesmata to understand cell-by-cell interactions, or looking to microtubules or actins that are linking with the shape of the cells. And also, more recently, we've been working on the stomata closure to understand using the autofluorescence whether or not how we judge a stomata is actually closed or not, because purely based on the guard cells, they're not correlated with the stomata closure at all. Obviously, these are linking with different treatment, different chemical, and also experimental settings. I don't want to talk in too much details about that. The second level, the tissue and organ level, we're using microscopy, and we're generating a lot of videos based on microscope. As you can see, I'm a computer scientist. I'm sure most of you are more experienced than me in this biological stuff. So we've been studied the callus deposition to understand how callus deposition were the pattern of callus deposition, and then linking that with lung immunity, and then generating patterns to understand the disease development during time. And more recently, we've been studying a lot on the smartphone-based analysis. You can see right here, basically using a smartphone totally offline without sending anything to cloud. You can walk in the field, take a picture on the plot from the overhead perspective, and then we've been using the YOLO 5, or more recently YOLO mobile net to detect the ears. We put all these things into an Android device so that you can take an image, and then using the computational power on the smartphone, you can generating a quantification like this within the unit area. So these can contribute into two parts. One part is obviously the traits. You can analyze this key yield components traits. The second part you can look into is within the unit area, how year by year this genetic gain can be continued through our work. But please, having an open mind, although these things are cereals, wheat and rice, it can be applied to other crops as well. It just, to us, most of these things are just mathematics. So if you give me an image, eventually what we're looking to is the features from these matrices, multi-scale, multi-dimensional matrices. At the organ level, we're particularly interested in how to do things in a dynamic approach. So you can see from here, this is using UNIT, taking images from the insect extrusion on rice. By training the AI model, this is basically by training the UNIT model to study which particular features can be utilized to define an answer. We could quantify the answer from these lovely panicles. And if we apply this approach onto time series, you can basically generating a good identification to understand at which particular time the flowering happened, and at which particular time the flowering reached a peak. And these are extremely important for rice, but once again, these sort of traits can be applied to other crops like barley, and also some of the flowering plants as well. From the detection of the deep learning models, we can generate things like that. It's basically the pattern to tell you when time goes on how different lines perform utilize the flowering as the trait. And the more interestingly is under different environment, particularly under different environmental factors in different sites, trial sites, how the similar lines can be compared in a quantitative manner. We already have QTL mapping on top of it, but because I just want to demonstrate our capability, I don't want to dig into too much. I will provide 2 case studies afterwards to explain to you guys regarding from images to genome to trait analysis, and then to the genetic mapping. Right. Another approach we've been doing is plot level. So Alec has been doing a lot on the in-field phenotyping using this sort of we call them selfie, like a camera system. It's just basically just a Bluetooth controlled camera to take pictures in a way that can be standardized, and then after that we could apply the YOLO method that I just talked about to quantify things like a panicle detection, or the wheat ear detection. On the top of it, if we manually select, at the moment we don't know how to select more appropriately, but if we manually select them, we can even extract the image out and look into the density of the spikelets for wheat, for example. And also to understand that during time, when time goes on, how these key yield components are changing. For example, utilize that, we can quantify the years, and then look into the grain filling procedure, the rate of grain filling during the period of time using this manual approach, but with the AI to analyze them. Another field level phenotyping was with LiDAR. This one was previously utilized on wheat, and then gradually expanded to maize. So you can see the work we've been doing is having this backpack LiDAR walking the field and generating the large scale point clouds represent the field. It's basically the 3D representation of the field. And then we built another algorithm, a platform called CropQuant 3D to analyze, to high throughput these point clouds. If from one field, you're talking about over 200 or 300 million points. And then we're generating a method to read these points, denoise it to get rid of all these outlines, and then eventually segment the plots so that we can look into individual plots, particularly at the canopy level, so that we can, using the dense 3D point clouds at the canopy level, which mainly contributes to yield. I think 70% of the yield coming from the canopy level, to generating voxelization, and then reconstruct them in 3D mesh, so that we can study and assessing the 3D canopy surface, which is highly correlated with the photosynthesis that we have been measuring in the field. So the paper has been published last year. Another approach, I will talk in details later, so I would skip this particular part, which is drone-based. I think Rob is quite interested, and quite a few people I've been talking with today, they are quite interested in the drone-based pipeline we've been building. So I will pay more attention to this. Our work not only focuses on research, but also looking to the applied research. So this was done a while ago with cheese growers. So cheese growers has been flying light aircraft in different plantation area, and then they generating this NDVI imagery. And what we have been doing is to using CNN model to study from this large NDVI imagery, how many lettuces can we quantify from the huge image. These images are like 5 gigabytes or maybe 6 gigabytes big for one area, for one plantation area. By identifying the number of lettuces, we also look into the density, the intensity of each signal of each lettuce, and to assessing them. Is this a big lettuce or medium lettuce or small lettuce? And then when we calculate the altitude that light aeroplane was flying, you have the resolution of the GPS, which means that you can getting the GPS out and understand how, Yeah, within the GPS grid, within one GPS grid, how many lettuce head I'm going to expect it to harvest. And largely speaking, within this small GPS grid, are they mainly big lettuces or medium or small size? And then we apply this work to a range of plantation area for experiments. The paper has been published, but a lot of algorithm that we are doing for this particular counting and trade analysis are open source, which can be downloaded directly from the GitHub. Excuse me. Right. Okay. Now this is the not the full spectrum, but a large spectrum what we're doing at NIAB, and also my Chinese lab as well. But I want to give you two case studies to explain to you in a bit more details regarding what we actually do from taking the data to analyze the traits to then linking with the genetics and also answering the biology questions. So the first one is was mainly based on the paper we published 2020 in New Phytologist, which we call them SeedGerm. It's basically using a Raspberry Pi, control the camera to take images. And when we're getting these imagery or image series, we analyze them through a combination of the supervised machine learning. So basically, the idea is very simple. Just get rid of the background and then extract the seeds out, and then basically monitoring how these seeds are germinated and utilize the cumulative germination rate. We're generating these curves or growth profiles, and utilize these growth profiles at a certain stage as the phenotypic variation, and then feed them into the TWAS, so that we can analyze the low size even with smaller population. Building on this, this was one of my PhD students that leading on this work. So building on this one, we have been establishing even more low-cost seed germination devices using a range of different devices, because we found that Raspberry Pi is getting more and more expensive. And the one approach to bypass it is using second-hand mobile phones, because mobile phones provide a lot of sufficient function we can utilize directly. As you can see, with this 50 pounds smartphone, which is like 7 years old, we can taking very high quality pictures, and then because they have all these internet, all these Wi-Fi systems, we can synchronize all the data directly with the OneDrive, which OneDrive helped us archive and manage these data sets on the cloud. And then building on the newly acquired data sets, we have using a different approach. We call them SeedGerm 2.0. This approach is basically training the data sets. Building the AI model is not hard at all these days. I have to tell you that even some students we supervise, like year 1, year 2 undergraduate students, they can easily build an AI model within, I think maybe a week, 2 weeks training. It's not hard at all. The hard part is actually the domain knowledge. To be fair, a lot of things we don't know what we are looking for. What kind of features is actually important for us to study and to extract from these large images. So the approach we have taken is to train them separately based on different organ parts. So seeds, seedlings, radicals, they've been trained differently, and then they jointly feeding into a multitask model, AI model, which means that we're using Jupyter to train these AI models, the deep learning models. You can see right here, this is time series from imbibition to protrusion to germination. And if I play like this, hopefully we can play it. Otherwise, oh no, can't play, sorry. I can't play this video, I'm sorry. This is not on my Mac, this is on Windows system, but I can describe to you. So let me go one step back. If you look at these tips, the radical tips, so basically these radical tips will be tracked using computer vision. This is not AI, this is computer vision. We track these tips I think a 4 hours interval. And then we can generating this basically reconstruct the radicals. So the color coding here is hours, from 0 hours to 71 hours. And then you can see that from the same genotype, so same seeds, we're monitoring at which particular time the radical emergency happened. And then we did not follow the actual image. We built the skeleton from that. It's a graph theory, basically, so that everything that we monitored based on a tip can be quantified. And we use this one to study not only the radicals, but also the seedlings as well, and then feeding them into some of the genetic mapping that Chinese Academy of Sciences are doing, to using the phenotypic variation to study rice domestication. Sorry, excuse me, what's wrong with my throat? And also looking to how the seed dormancy is linked and can be represented by the phenotypes. So from image series to trait analysis to genetic mapping to important loci that could be utilized for breeding or for biological discovery. I added in a little bit more after talking with Fraser. This is our multispectral seed imaging system. So you just saw the seed germination system. Now this is more about hyperspectral imaging. Spectral traits can be utilized for genetic map, for dissecting the genetics behind important biology questions as well. So you can see right here, we built this system called, we partially purchased a system called Videometer. And then we've been utilizing this conveyor system to scale up the throughput of this imaging. So what happened is, when we're using this one, when we're using this device, we can generating 22 channels of bandwidth, and some bandwidth you can see right here. The bandwidth basically is from 375 nanometer to 975 nanometer, and some bandwidths are highly correlated with seed vigor. Some bandwidths are highly correlated with the chlorophyll involved in the seeds. And I think for people who are doing seeds, they will understand that the chlorophyll in the seeds actually interfere with the longevity of the seeds. And if I taking some of the spectrum out, you can see that we can quantify directly with how these seeds will look like, whether or not this seed coating has been damaged during the shredding, or we can look into the seed uniformity based on certain the nutrient distribution, and also some of the seed vigor that we've been studying. I can explain to you what do I mean seed vigor in this particular case. What we have been, the approach what we have been taken was to get rid of the background as well, and generating a method. This is actually quite complicated method to loop through all the images, all the seeds that we are having, and then if the seeds are connected, we find ways to divide them. This is a combination of the superpixel approach together with the feature extraction approach. But anyway, you can see right here that through this one we can divide all the seeds into individual seeds within about 10 seconds on a normal computer. And then we're using each seed as the region of interest, and then overlay going through the spectrum that we know will be useful. So you can see right here, these are the reading from each individual spectrum. So what does that mean? The first one is, by remove the background, we can look into the morphological features from these seeds. So I'm sure this is very important when you harvest for barley or for all sorts of seeds that you think you want to pay more attention to the research on. The second part is, you can see right here that we have been picking for the spectral bandwidth, basically. So ultraviolet, blue, red, and near infrared. You can see the ultraviolet linking very closely with the nutrient density. Blue is chlorophyll, red is oxidation, excuse me, and the water content is the near infrared that we are utilizing to study. So what do I mean bigger afterwards? This is basically when we combine different bandwidths together, not only single individual bandwidths, but a range of bandwidths together. We can measure, not all the seeds can be measured. It's for the embryo, by the way. It depends on how the seed is being oriented, but it doesn't truly matter. If you have 100 seeds, you measure 50 seeds, it's enough for you to get a very good understanding about the embryo size from these hyperspectral imaging. And then you can cluster them. You can put them into the quality clustering method to understand, utilize the combined the morphological features with the spectral features together with the seed vigor features or embryo size, that we can divide them into different classes and hence the approach for seed testing. We even put these seeds, different class of seeds, into the field to study their seed, their early establishment, to understand how these traits are linking with early establishment directly. But this method, or this hyperspectral or multispectral imaging, is not only limited in seeds. We can apply them onto leaves as well. You can see right here some of the things we've been studying, and I think this could be really interesting for Fraser's work. One thing that we've been studying is to look into the coexistence on yellow rust and the Simo septoria. And you can see that based on different time, you can basically look into at the latency period before the minor symptoms can be seen, whether or not these spectral changes can be detected. So that was one thing we've been discussing downstairs in the imaging room, that we found, not only we found, but also other people found as well, that from between 500 to 750 nanometer range during the latency period, the spectral signature is jumping from healthy plants to affected plants. But the problem is, if we fly the drone, we can only see things from overhead. We can't see things beneath the canopy at all. And another thing is, we found that biotic and abiotic stress cannot be differentiated at early stage. So whether or not these are caused by drought or heat, or this is caused by disease, we don't know. But the thing is, it's a good indicator. It tells you the plants are not performing as they supposedly performed, and hence the possibility to do investigation. But don't forget one thing: septoria has 15 to 21 days latency period. It's a very long latency period, and also the spectral is changing, which means that it's a good indicator for us to study things in the field and to do something to prevent some of the things happening. Right, okay. Now we're talking about field. The second case study I want to talk about is area imaging. As you can see right here, let me briefly explain to you guys. It's very good to see the Sebastian and Paul's work on the Germinate. And we've been doing very similar things as well. When we have in field experiments, we we designed experiments, flying protocols to study how to image the plants. It's not only 2D on the MLX, which is the 2D representation of the field, but also the 3D reconstruction. So basically, we're generating a lot of point clouds to represent our crops in the field in 3 dimensions. And because we have the spatial information plus the spectral information, what we can do is combine them to generating different kind of traits, like a morphological traits, which is a plant height, canopy coverage, vegetative indices, looking to green leaf area, VAI, which is plant health, NDVI, NDY, which is yield potential, and also the texture traits, which are particularly useful when we assessing the lodging. There's loads of lodging in Bali, by the way. We've been looking to field trials, and we were astonished. But anyway, you get my gesture. We built this this pipeline, and this pipeline basically has been published on New Phytologist this year, which is combining 3D and 2D to study the traits. But this is one part of the story. As you can see right here, I try to briefly explain. So from point clouds, we denoise them, and then we find which particular point clouds can be utilized to be treated as ground points, so that we can divide the ground and the plants, and then utilize this spatial information of each plot of the plants to study the traits. Yeah, you can see this is basically when you fly a bit lower with the drone. Then the color coding is basically the height information. The higher, the redder, the more redness they can gain, and the lower the blue. Right. Once again, I can't play back this video now. Sorry. This is one of this is basically when we're generating 3D point clouds, we can mimicking how the drone is flying in the field, and to show you from very high level to very detailed level, plot level, how the plots are looking like. It could be a good way, fantastic way to be the database in the future. If you want to see at one point how your plants are performing in the field, you can basically utilize these 3D point clouds as the data sets or database to support that. It's fantastic. It's moving. I can drink a bit. This is basically, as you can see, that's the nursery plots we've been planting in this season. Not this season, but the 2021 to 2022 seasons. Yeah, if you can basically mimicking how your drone fly in the 3D scene, and if they're lower, then basically you can see a lot of morphological features. But because this is just everything is generated by a drone, you have some side missing, some side points missing from the video. This was done like before the summer, and it's a good way to now having like a database, like a 3D database. You can mimicking how you get into the field and analyze them. Based on this work, we have been expanding to much large scale. So these are 2,000 yield plots that we have been working together with RAGT. As you can see right here, that's the fields. So when we fly the UAVs, we're generating these point clouds for the 3 fields. And then we feed them into, based on a GPS location, we feed them into the AirMeasurer system. So this is the paper we published during the summer. So GPS location tells us the region of interest, and we extract them out of the region of interest, and then apply, and then based on the morphological features and the spectral features, it divides all the plots. So each individual plots will be divided by the software, and then you can apply batch processing to study each individual plots, how they perform during the season. What do I mean is, if you have a very well designed field trial, each row here is when you fly the UAV, each column here is this particular genotype perform visually in the field during the time. And you can basically organize these lines based on genetic background, so that you can combine this biological replicates next to each other and see whether or not they perform exactly the same, either at one site or at different sites, and hence the comparison. So because we have this sort of key points, key representative points, performance of the crops, then we push this one a bit further to generating a dynamic analysis for traits. All of our traits are dynamic, to be honest. Nowadays, all the low-hanging fruits has gone. It's important to understand how to look into these dynamic traits to study things like stress resistance, to study things like in the context of global climate change, particularly when plants are interacting with the environment. What is the dynamic performance? These traits we're interested, agronomic traits or the traits that we've been selecting, are interacting with the environment and hence to study what kind of genetics underlying these traits. So you can see as you can see right here, each dot is when we fly the UAV, or the represented points I was talking about, which is linking directly with the matrix, the performance matrix here. I'm using height as an example. There's plenty of traits you can utilize exactly the same way. And then we're fitting a model on top of it. We're using Gaussian model for height, but there are different models for different traits as well. Depends on how they're performing. And then we study the first derivative, which means how this particular trait is changing during time. And then, as you can see right here, the two key points, we call them key knee points. This knee points basically is telling you at which particular period of time that this trait is increasing. And then within that range, we define the rapid growth period for height is the trait. And then we look into the second derivative, which means that how quick the change has been changing, which means that at that particular moment for the height trait, there must be the most active gene regulation, gene expression to regulate that particular trait. And when we utilize that, when we define this sort of fastest growth rate together with the rapid growth, we basically defined this trait in a dynamic approach. You have to think a bit further. It's not only about performance. If you're looking to like a disease resistance, it's the same idea, the same mathematical idea. And then we apply them onto seasonal trait analysis. You can see right here on 3 different kinds of rice subgroups: Japonica, Indica, and intermediary. And the study very similar approach. You can see this is not only height now, this is the canopy coverage, and the other one is the excess greenness, which is a green leaf index. And then the genetic mapping, the QTL mapping will tell you the story. So if you're only using maximum height as the static trait, then you can get in this semitopin gene. In rice, we normally have to hit this particular locus before we believe that the result. And the GHD 7.1 is the stem elongation. And if we change from the static trait into the growth, the dynamic trait, you can see it can be high growth rate. It's not only we're hitting, it's not only hitting the SD1 or GHD1, we're also hitting something different, which shows that the rapid growth phase has certain loci that can regulate that. Obviously, as a computer scientist, or particularly for phenotyping perspective, our boundary stops here. As for how to make sense at a gene level, it's not my job. It's a collaboration. We provide the weapon, the enabling technology to support biological scientists, not saying that we are doing this gene level work. It's more, but there are methods we have been utilized. I will talk about very soon in the next 2 slides. We'll talk about how we apply them onto the nitrogen use efficiency as well as nitrogen uptake. This is more about growth and development, so performance-related traits. You can see that if I apply them on the canopy coverage growth rate within the sort of maximum coverage days. Not only TEC1, the famous gene on canopy structure, can be detected or can be hit, but also we have seen 3 other peaks. As a matter of fact, Chinese Academy of Sciences, Professor Bing Han's lab have been longing to look into why TEC1 is not making sense on some of the canopy closure. And then they found that actually there are other peaks within that region as well. And it's highly likely that the major genes that regulates the canopy coverage closure has other places. But once again, the computational method, the mathematical method, is try to help people sitting here to open a new door to understand and dissect genetics. when we are not saying that our work is the end point. Our work is only the enabling point to help you guys to advance your research to the next level. Similar things can be seen directly on the GWAS analysis as well. I mean, I don't want to talk too much about that. As promised, I want to give you a bit more details regarding how to dissect traits. Previously, it's impossible to send people in the field. If you apply nitrogen at a tilling point, you're going into the field, you can't really be there 30 days every single day. And hence, you can't really getting all these traits out that are highly correlated with the nitrogen uptake, for example. So utilize the dynamic phenotyping, we started to understand how to dissect the traits because we can build in this model. As you can see right here, these models are using different algorithms, not only Gaussian, but also they have using linear regression, and they're using moving average, and also some other traits. I'm only using some traits here to demonstrate. You can see the growth curve here is correlated with the images right here. These images are real images captured in the field. And we use this one as every 5 days is one interval. And these five days intervals allows us to understand that how different nitrogen applications have been taken, have influenced the trace changes, like height changes, coverage changes, or ASM. ASM basically is the canopy structure, how the 2D canopy structure is changing. If they have more leafy, or if they're kind of pointy, you can always using texture to quantify that. By doing this, I think, do I have the next one? Yeah, okay. Then you can collating all the traits you are having and put them into the PCA, 3D PCA, to understand which trait are contributing to the highly nitrogen efficiency lines that you will be interested. So this is just like a simple model, which is basically transfer from multiple traits into a number of key traits that you would like to utilize to analyze nitrogen uptake during the period. And this x-axis and y-axis or z-axis are all different. These principal components are different. Sometimes they are highly linking with plant height and the canopy coverage. Sometimes they are highly linking with the green leaf area, green leaf index, or other canopy structure like ASM. But as you can see, very interestingly, by 3D grouping that using the highly correlated traits, you can divide, each dot here is one variety that we've been studying with 3 replicates. And you can see after nitrogen application, it started with 2 different groups, and then it gradually group in, and we found one line here, which is highly responsive. And then when the time goes on, it's still highly responsive on the, I forgot to put it, this is 1 to 5 days, 6 to 10, 11 to 15, and 16 to 20 days. And and eventually everything grew back again. It's like the plants are taking nitrogen, taking resources, and then they develop for a certain time, and then they stopped, and they kind of like digesting a little bit. So as a computer scientist, I don't know how to describe them in much more details, but it seems that there are loads of hypotheses can be generated through that approach. We even utilize this 5-day differences for 5-day differences. When I say differences, basically it's growth changes, growth rate, to do this genetic mapping, to understand what kind of genetics underlying the nitrogen uptake. You can see right here, this is 1 to 5 days, 6 to 10 days. As I've been explaining to you that all these loci, they're expressing differently. So sometimes in the first 10 days, they express at a certain level, and then they gradually were kind of faded. And then some other loci picked up and started to express as well, which I would say I haven't really think that dynamic phenotyping can be utilized in this approach, which is basically telling you that what is the loci of interest or locus of interest that can be dissected through the dissected traits, which I think, once again, has opened a new door for us, which we could really look into and make much more precise genetic mapping or genetic mapping studies to linking with genetics and also the molecular markers that we're building for breeding or for research. Right, okay, I know time is very limited. I'll quickly talk about the third case study, which is more about other things, the data for data sciences or data analytics work we've been doing. One thing is crop growth modeling. If you have loads of data, particularly with the environment data, you can look into G by E by M with the agronomic management data, and also some of the I should not talk about root stuff at all, but well, we do a bit of root analysis as well. These are very much open source data sets. We will be happy openly share with you guys. We also do a lot of Apple Ultra stuff. So generating 3-dimension point clouds to understand at a tree level how can we look into the blossom cluster, as well as quantify the apples on these trees. Something very, very low cost using the structure from motion algorithm, taking images from different perspectives. Basically, the strawberries changing orientations, and then we reconstruct them into high resolution, and then basically analyze them from this high resolution 3D strawberry models. And more recently, we've been working on nematodes detection with the Cambridge University as well, with Sebastian even is then the group. I never call him this way. I just call him Sebastian. Yeah, so basically using AI to detecting the nematodes. I think you can see right here we're using a combination of the instance segmentation plus the semantic segmentation methods, and then you can see that from very complicated background, how can we getting these nematodes out and quantify these nematodes with the morphological and also the spectral features. You can see on different experiments, these nematodes are big. Even the background is very, very complicated. We can still easily detecting these nematodes. If you have a chance, if people are in the computer vision area, you will see that this is extremely hard to analyze these nematodes because the background is so shining. I mean, I'm sure Fraser knows that. But this deep learning method, by looking to not only the semantic location issues, but also looking to the instance, we can fit them out. We can segment them out. And the last thing that we've been doing a lot, which I think Rob would be interested, is AI predicted method. This is basically can be expanded to much bigger application areas. So one thing is, if you have RNA sequencing data, transcriptomics data, you have loads of gene expression data, which means that all these gene expression data can be treated as one element in an image, in one image. And if you transfer the image, so each dot, each element on this picture here is one gene, and the gene expression is the intensity, then you can link the image with the outcome. The outcome can be severity levels, so that you can building the relationship between the genes to the final stages of disease or severity of disease or other things that can be quantified by domain knowledge. And if we basically reshuffle this one for 1,000 times, the result you're getting is very reliable. If you reshuffle the gene, because if you think that one image has just one by one relationship, then if you reshuffle and you hit this gene again for 1,000 times, then it's very, very precise and basically tells you that this particular gene definitely contributes to the severity levels. you want to modeling or predicting. We've been using permutation and the GregChem. GregChem is a reverse engineering on the deep learning method to score. So basically, we have two scoring systems of the gene of interest, and then by combining the permutation, which is more like a traditional statistical approach, with the deep learning reverse engineering approach, we can generating the joint list and then putting them into the goal the enriched goal terms to understand their goal function, gene function, and also eventually getting the regulatory network out of it. This particular part is very similar to the GenE3, which has been previously published to understand how these gene nodes are connected, and hence helping with biology experiments to understand what to knock off, what kind of connection or relation between these gene functions can be studied. Right, okay, I'm glad that I can finish before 3 o'clock. This is basically what I want to share with you guys. One part is basically to demonstrate the capability from my department. The second part is basically to convey some of the case studies that we have been carrying out at my laboratory. Right. As all these Cambridge guys normally do, they put in these Latins that nobody knows. Time. I think, well, we are facing to a certain point that a lot of technology can be applied to enabling our biological discoveries, and I'm happy I can be part of it to help you or to collaborate with you to make that happen. Right, acknowledgement and collaborators. Thank you very much. Thank you very much.
Thanks, Ji. Huge amount of work and a lot of different applications. Does anyone have any questions for Ji? No? Okay. Well, I'll start. So obviously you've got a lot of different things going on there. There's a lot of work going into it. Are there any specific bottlenecks, and are they common to all of those different approaches, or do you find that different approaches have different issues in terms of resources and resource management?
It's a very good question. I do mean it's a very good question. The thing is, when we were involved previously, it's always people done some work in the field and say, well, there you go, that's the data, make some meaning for us. I don't think computer science or computer vision or trait analysis, phenomics will make sense from that perspective. It's important to involve your data scientists at early stages. It's important to understand what's the requirement, what kind of traits you will be targeting, and jointly design an approach that can be followed during the experiments. So involving your data scientists as early as possible. Even they might not talking the same language, but it's important to convey science towards both sides. Yeah, so I guess that's partially answer the question. The second part of the bottleneck obviously is the domain knowledge, because the domain knowledge will make a huge difference in terms of shaping the algorithm that we're utilizing, shaping the solutions that we would like to develop.
And I guess a related question is, I assume that this is very data heavy, right? Do you need an awful lot of data infrastructure to be able to deal with this, or?
I think that the Germinate here is a fantastic infrastructure. We would like to try it as well. And we using data infrastructure basically. I think that they are project-based. So different project, if they come in with different data sets, we normally have either file system or a data management system. It's a little bit different. But once again, I think gradually with the UK phenotyping infrastructure in place, we will having a national level metadata plus the data structure that everyone can utilize. I think this could be helpful in the future.
Do you have any questions from the people watching online? Feel free to unmute. Or Donna, if there's anything in the chat box, I can't see it from where I am.
There's no questions in the chat box.
Raoul.
I'm curious, so we are moving from high-throughput platform to phones, to smartphones. So I have two questions regarding that. The calculation is done on the phone, or the phone is as a tool to send a photo to a specific place to do the analysis?
Depends on who developing these things. So, for example, I was showing Rob yesterday when we were having dinner that nowadays we can put in the very heavyweight smart AI models onto your smartphone. I can show you later that taking an image of the wheat spikes, wheat, and then it quantify the spikes for you. But you need to using Android system and AI model to be connecting with the CPU and the GPUs directly on your smartphone. If you're just running everything purely based on CPU, it would be very slow. And also, if you think about applications in the long run, if you got a chance to go to African countries, go to say Tunisia, for example. I was in Tunisia during the summertime. There's no mobile network. You can't send things to cloud and waiting cloud to coming back with computation. And for farmers, particularly that Gates is supporting, that low-income farmers, they do have smartphones. Nearly everyone got a smartphone. Take an image, run it on a smartphone, coming back with the yield components or the traits you want them to understanding, like a variety recognition. It will resolve the problems that most people will be asking. I don't think they're asking biological questions that intensively, but they do want to understand certain traits directly from an image. And some traits don't need to be run on your smartphone because, for example, I think I was talking with Sebastian, if you're using a ruler, getting into the field and measure the height, it's faster than your smartphone. But if you want to quantify the leaf area, the index, or the sort of within the unit area, how many spikes you can quantify, that take about 5 minutes, even by experienced people. Then the computational stuff will make sense.
Second question, sorry. For the tracheotomy data, what kind of data do you need? Are you using the raw data, or or someone need to provide you any kind of filtered information?
You mean
for the gene expressions?
Oh yeah.
Sorry.
You need to normalize that. It can't be analyzed directly because it's skewed data. You have to normalize it to make sure that the level is within the range. Otherwise, it's rubbish out. Excuse me.
So when obviously shown that you can capture vast quantities of data with drones, your sort of benchtop multi-step system as well, how do you find yourself ground truthing that? Is it still predominantly by hand or by folk out in the field with their rulers, or do you try and then deploy the models you're making to reinforce and learn the ground truthing data to then doing the reinforcement learning as well?
Ground truth is always happening in the very beginning. We will have to have domain knowledge guiding the results. There's no doubt. But coming back to more data sets, for example, the GWAS analysis that we're talking about is pretty self-explaining the validation. If your menu counting is making sense, correlation-wise, you can start square, you can see the RMSE to understand how the deviation from your computational data and the human data in the first season. And then on the second season, if you want to study the things like genetic gain and other things, for example, then your phenotypic data year by year will tell one story, whether or not it's correlated. Obviously, environment might be changing, but using dynamic phenotyping, you can look into the period of time you want to study. So that the big data will give you the correlation and the validation as well. Yeah. So does that answer your question?
Yeah.
Yeah. So it has to be 2 tiers. One tier is the human data for ground truthing. The second tier is actually computational data itself, whether or not it can be repeated.
Can I ask what some of the challenges are with working with dicots compared to the monocots in terms of things like plant height and canopy density and things? Do you find it a lot more challenging to do the lettuce and the strawberries and the turnips?
We don't do strawberry on plant height. We're just looking to the strawberry quality trait. We found quite challenging when we apply the wheat model to the rice, because rice lodging a lot, particularly if you're looking to land races. And it's even more challenging when we apply them to barley. These plants keep on lodging. Yeah, so maybe that is the best. That is more about when we get into the analysis, when to analyze. So, for example, if we want to study ears previously, we can, for wheat, for example, we can wait until maybe the middle grain filling or even later. But for rice, we often go inside when inside the field around, say, flowering time, or even a little bit like early grain filling. We found that barley, early grain filling is a must. A bit later, it's all gone. Yeah. So I think it's timing. It's always timing.
Okay, so no further questions. I'd just like to say that it looks like hopefully there'll be quite a lot of collaboration going on between us and you guys. I'd just like to thank Ji one last time. Thank you.